But one of the things we used to worry about is, can microbes actually survive in these extreme mining environments? And so we used to do things like plate counts or bug bottles. And many of you on the call, I've saw some of the names. I know you're familiar with those processes. And what we found is less than 1% of microbes like to grow in a bug bottle or on a plate in the lab. We just don't do a good job of giving them what they need to survive, especially replicating these extreme environments like we have in mining. So we can't give them, the right pH or the right temperature, all these things that can be important. So we used to think they didn't survive and they weren't present in these environments. But with the use of molecular tools like we have today, we're actually able to understand that not only are microbes present in these extreme environments, but they actually, some of them really thrive under these conditions. So organisms like acetophiles can actually thrive in really low pH of 2 to 3, or sometimes even lower. There's an organism I'm going to show you today that was surviving at a 1.5 pH. And then there are certain groups of organisms that really are not bothered by high metal toxicity like other organisms are. So they can help us with these high metal concentrations that are present in different locations. A group of organisms that we really need to become familiar with in mining are called chemolithotrophs. And these are organisms that gain their energy either by oxidizing or reducing inorganic compounds. And as we know in the mining environment, these are extremely rich in metals and in minerals like iron sulfides and other elements that actually make a great home for these organisms to be able to live and to thrive. So in these environments, we'll see organisms like Acidothiobacillus ferroxidans, and this organism in particular can not only live and thrive in the mine, but it can help us extract metals through a process called bioleaching that we'll talk about today in this presentation. There's also organisms like Leptospirillum and Thiobacillus that sometimes might be a cause for concern, as they're key players in the formation of acid mine drainage because they can oxidize iron and sulfur into sulfide minerals that will eventually result in sulfuric acid. But if they're present in certain parts of the process, they may actually be helpful to us because they can actually be utilized to help extract metal from our ores. And not all organisms are doing something bad in our system. Microbes, just like in our the human health, there are bad microbes that can cause problems and cause infections and diseases. There are also good microbes, like in our gut microbiome, that help us with digestion. The same thing happens in mining and in remediation, where we can use these processes for the good of what's happening. So in situations like this, we can look at sulfate-reducing bacteria who will produce sulfides that can help us precipitate dissolved metals. So we can use the SRBs not only for the change that's happening in the sulfide formation, but we can also use it to precipitate the metals that we want at different parts in the process. And then there's organisms like arsenite oxidizing bacteria, and they'll take arsenite and change it to a less toxic arsenate. So if you have that in your system, looking for these organisms can be very valuable. And then there's jarosite producing microorganisms that can also help with bioremoval of arsenic. They actually adsorb this compound and this harmful metalloid into the microbial cell. And we'll talk about that just more in a minute as we look at different processes that microbes can do. And when you think about bioabsorption, this is very cost effective. These organisms are naturally present. They naturally have this capability for bioabsorption to happen. And so it's a very environmentally friendly alternative to some of the traditional, really invasive methods of recovery that we use today. So we can use less solvents, we can have less manpower that's involved in this if we harness the power of the microbes and start to truly use those better. Another tool we can use that microbes possess is something called bioaccumulation, and I mentioned this with arsenic just a minute ago. There are microbes that will naturally produce organic molecules that change the oxidative state of metals, or they chelate them, and that makes them bioavailable to the cell. So as this happens, the cell might actually uptake some of those metals and retain it inside their cell walls or in the cell wall itself. And this happens differently for different organisms and to varying degrees. But through great research, we're starting to learn which compounds that they can chelate and utilize and which ones are kind of held within the cell. So that can be beneficial. And when we get to the end of the presentation today, I'm going to show you some of the future of how I think we can use this. So finally, we have the ability to utilize bioleaching to help us for recovery. So as microbes release these organic compounds and the metals solubilize, the rare earth elements and other metals can actually be released into solution. So then processes like precipitation or solvent extractions or ion exchange can be done to recover them better from solutions. And you can get a lot better recovery rates than you can without the bioleaching being involved. If you've heard me talk before, I've said this word a lot, it's quantitative PCR. So qPCR is what we do now to count the microbes that we're interested in. So we can look at a microbe by name, or we can look at a group of microbes and what they do, like sulfate reduction or iron reduction. So when we tried to do this before with culture, as I mentioned, we'd have to take the sample, ship it to the lab, try to replicate the conditions, grow it on a plate, and it was just really unwieldy to be able to do it. With DNA-based technology now, we take the sample from the environment, extract the DNA from that sample, and then we use something called polymerase chain reaction, or PCR, to amplify an organism or a gene that we're interested in, and we can count how many of that we have in our original sample. And the pandemic really brought about the value of qPCR. We saw with COVID identification and quantification, it was a common household name. So now I don't have to describe this process as much as I used to. We've been using it in the medical industry and in environmental remediation literally for decades now, and it's a very valuable tool. But now we've proven time and again that this is the most accurate way to quantify different organisms and genes. And then the other thing that we can do, so now we know we have this valuable tool, and I can tell you how many sulfate reducers you have in your sample. But what if, like in mining, we need to look at a lot of processes? So I want to look at ammonia oxidizers, and I want to look at manganese oxidizers. We have a platform called QuantArray that you can see here. So this is a slide that has a square, and it has a lot of little holes. So then we can do qPCR reactions on a very small volume in each of these holes. So we generate quantitative numbers of each of those organisms or processes that we're interested in, so we can do it all at one time on a sample. So this gives us a lot of information that I'm going to show you in our system. So we can go in and look for very specific organisms that we're interested in. And some of these, like arsenide oxidizers, we don't have assays for yet. So that this can be a very beneficial tool for us to use. It's also really, really beneficial for us for rare earth element recovery. What we're learning is this bioleaching of certain rare earth elements is better with certain genera of organisms than others. So with some bacteria and some fungi, we can start to bioleach different elements that we're interested in, like a monzonite, or different light or heavy REEs. We can start to do better bioabsorption, bioaccumulation, and bioleaching by knowing what these microbes can do. So if we do a sequence and we see that we have a lot of this micrococcus that's present, that means we have a really high likelihood for bioabsorption of heavy REEs that's happening in this particular sample. These are our nitrogen fixing bacteria. And this group is very important as they can be easily inhibited by heavy metals. So we were worried about that because metals will actually inhibit the activity of the nitrogenase gene that they use for fixation. And here we see that not to be the case, right? We have a good concentration around 10 to the 4 at the inlet, and at the outlet we have almost 10 to the 8 cells. So it doesn't seem to be a concern at all in this situation. And they're not only present, but it looks like they're thriving there at the outlet of this area. So nitrogen-fixing bacteria are really important in this process because they can remove heavy metal ions from solution. They use biosorption, and they can also help us to precipitate these heavy metals in general. That's exactly what the goal of this holding pond was. So for the last 25 years in my career, I've really been working on trying to develop a field extraction method that was easy to use, that didn't require equipment in the field, that provided us with good, high quality DNA that we could use for assessment. So we now last year started offering something called the bioextract, which we can use in site locations. We're using this in many different countries around the world now, in remote locations. We also used it in this particular situation to help us overcome the radioactivity, because what we could do is they had 5 metal coupons and they wanted to look at this cycle every 45 days for 4 cycles to see if corrosion was increasing and what we were seeing colonizing these coupons. Well, since we couldn't extract them in the lab, they were able to extract them in the field in about 45 minutes for all of these coupons and to get the DNA, so then we could analyze the QuantArray, and this time we did the QuantArray MIC for them. And so it was very exciting, and what they did was measured, they looked at the radioactive concern in the DNA after extraction, and I've seen this in working with universities around the world. Once we extract the DNA, we've removed that radioactive label that is there, and so we're able to then ship that sample and have pure DNA that we don't have to worry about.